Hello and welcome to NanoReach, a scientific outreach podcast by the Nanomedicine Research Group at the Institute of Chemical Technology, Mumbai. In this episode, we have Aditya Narvekar, who is a PhD student. He talks about his past experiences and his current research. Let's begin. Hello, Aditya, and welcome to the NanoReach podcast. I'm so happy to have you here, and I'm really looking forward to a conversation today.
Thank you so much, Parth, for inviting me for this podcast. And it would be great to chat about science in general and also reach out to the masses.
So, see, the main intention of having you here is to discuss your research and how it can be useful to the society as a whole. But before we go there, I wanted to ask a bit about your background before joining PhD. So, could you tell us
Yes.
about your educational background and how did your experiences during those years motivate you to do a PhD?
Okay, yes.
Yeah.
So, as the similar story goes, my CET score was not so good to get a medical seat in Mumbai. So, and I didn't really want to go out of Mumbai for MBBS. So, I continued to pursue my degree in basic science. So, I did my BSc in microbiology from Ruya College. And during my bachelor's, we were encouraged to do several summer projects. So, after I finished my second semester of the first year, I had got a chance to work on microfluidics. So, where we were trying to replicate one of the basic bacteriological techniques, which is enumeration of bacteria. So, enumeration is generally done by spread plate method in regular microbiology labs.
Yeah.
So, there we used to try to correlate the spread plate method results with paper microfluidics enumeration results and try to make the procedure more economical. So that was the start where I really got interested in doing something apart from just textbook learning. And thereon I continued doing several projects during my bachelor's. So the next project which I did again during my bachelor's was a part of my second year work, which everyone was supposed to do. So it was a basically a compulsory thing which we did in the Department of Microbiology.
Okay.
So we have an annual festival called as Microbes, and we are divided into several group of students, and we are supposed to carry out some research activity. So the theme for our microbes festival was microbial interactions. So everyone was supposed to carry out their work on various interactions in nature which occur between any microorganism.
Okay.
So we were focusing on a particular microbe which is established for consuming plastic in general. So the name of the microbe is Pseudomonas putida. So Pseudomonas is known for degrading plastic, and we were trying to increase its capability to degrade plastic by its interaction with another microbe, which is Saccharomyces cerevisiae. So what we did in that project is that it was very basic work because we were just second year students. So we tried to take the secondary metabolites of the Saccharomyces culture and tried to grow Pseudomonas in the presence of that secondary metabolite. And we saw that Pseudomonas was able to degrade the plastic in a better way in the presence of Saccharomyces as compared to doing it alone.
Oh.
So we tried to show this microbial interaction during our project work. So that was another project which I did during uh, my bachelor's degree. And then in the third year of my bachelor's degree, I was working on another, which was a voluntary project. This was not a compulsory activity. So we had got several contaminants from a fermenter from a lab in IIT Bombay, and we were supposed to identify those contaminants as a part of our research work. So, basic advanced technology identification is based on 16S rRNA sequencing, which is the most conserved nucleic acid amongst all bacteria. And but traditionally, this work was carried out using Burgess manual. So we tried using the traditional approach. Using the Burgess manual, we carried out several identification tests and we identified the bacteria for them. And then they tried to correlate that with 16S rRNA results, whether they really, match with the identifications done traditionally. So
Okay.
this was a very fulfilling research work experience during my BSc, which really rekindled my considerations regarding what I should pursue ahead and continue in research.
All right. Yeah,
Joined.
I just had one clarification. So the
Yes.
Burgess manual is the thing where you start off with gram staining and you
Yes.
divide,
Yes.
right?
Yes. Yes. So
Uh,
you carry out basic characterizations test based on the cell wall, and then you do oxidase test and IMVIC
right.
test and
Yeah.
so on.
Yeah. Yeah. So basically characterizing the different types of
Yes.
microbes.
Yes. Yes. We tried to identify till the genus level, and then we further went on to the species identification as well. So
And after this, you did your masters in.
I did masters in biophysics because I was really interested in protein structures and how they function.
Oh
So
wow.
this was the subject which really focused on proteins a lot. So I went on to pursue my MSc in this subject from University of Mumbai, and there the since the subject was new during the first year, I didn't really carry out any extra research activity. My more focus was on strengthening my theory about biophysics. And during the second year, as you know, that all master students are supposed to carry out research work. So I did my MSc work dissertation project in the Center of Excellence in Basic Sciences in the university itself with Dr. Avinash Kare, who works on actin depolymerization. So his lab basically works on ability of any external agents which are able to depolymerize actin, and this research is basically extrapolated to the ability of stopping any medicine or any nanoparticle from not allowing the malarial parasite to multiply.
Oh.
So if you stop the depolymerization of actin, you basically stop the multiplication and the cellular integrity of the malarial parasite. So the extrapolation of research was towards malaria. So my work was basically to see which nanoparticles are able to do this actin depolymerization activity. So we basically extracted the uh, protein from the rabbit skeletal muscle, and because actin is a very conserved protein,
Yeah.
the rabbit skeletal muscle actin resembled with the human actin up to 72%. So we could go for an animal source, and we extracted the protein and then carried out its polymerization and depolymerization, and see whether using fluorescence based experiments we tried to understand whether these agents were able to depolymerize actin or not.
Okay.
So that was about my MSc work.
And how did your masters, convince you to do a PhD, and how did you get to know about NRG?
Okay.
Huh.
So until my like masters got over, and since I was doing it from the university,
Yeah.
I had to really wait for the results to be declared, because the university declares the results of a lot of colleges.
Hmm.
It's not autonomous. So I had a lot of time between my result declaration and my exam, which got over in June.
Hmm.
So I had to take up some position which would really bridge this gap between my result time and the exam time.
Hmm.
So immediately after my exams of MSc, I went on to do a junior research fellowship work in ISER Bhopal. So where I joined Dr. R Mahalakshmi's lab for which basically focused on membrane protein folding. So she basically tried to understand the folding and the unfolding process of various bacterial membrane proteins, and the folding unfolding process will basically tell you at which stage of the folding process the protein is weak, and you can target the drug towards it so that the bacteria loses its viability. So her work was basically focused on that, and I worked on a particular membrane protein called as ale,
Called as?
which which was a membrane protein from the cell wall of the bacteria called Yersinia pestis.
Okay.
So we focused on the membrane folding of the membrane protein folding of this particular protein, and we were trying to establish its folding mechanism in the presence of other proteins which assist the folding of these proteins in vivo. So we were trying to replicate that in the in vitro conditions, whether the folding mechanisms are correlating.
Okay.
So until I finished this work, it was almost the time when I got my TSD Inspire Fellowship for pursuing PhD, and also I got my MSc results. So after finishing this work, I came back to Mumbai, and I started appearing for several PhD interviews. And during the course of giving PhD interviews and several non-successful interviews, I ended up here.
At
So,
NRG.
yes.
So, what's your research at NRG aimed at?
So basically, as you know, that in Nanomedicine Research Group, we work on several areas, and one of the areas is biopharmaceutical development. So, as you know, that biopharmaceutical development is a well-established process, and there are several steps which lead to the final biopharmaceutical product, which is ready for patient use. So
Okay.
it starts from cloning, and then downstream processing, and upstream and downstream, and finally you characterize the protein, then formulation development, and then the regulatory approval, etc.
Right.
So my work is basically focused on the characterization of these biopharmaceuticals. So when a protein is being, you know, uh, purified, and then you characterize the purified protein for its various attributes which define its structure and define its size. Now, these attributes are very important because, as you know, there is a structure function correlationship. So, until and unless the protein is structurally well formed, you can't expect it to function properly.
Yeah.
So, that is why we have to be very sure about its various physical and chemical characteristics.
Okay.
So, when you're characterizing the various aspects of these proteins, you basically try to understand its physical and chemical structure, and using various techniques which are high end, mass spec, circular dichroism, and so on.
Yeah.
These are all scientific terms which are denote the principles of these techniques, and even HPLC or dynamic light scattering, analytical ultracentrifugation, and so on. They are used for characterizing these proteins. So the need for using so many techniques is that you try to confirm at each stage that what you get from the previous technique is really correlating with the next. So although the techniques are principally different, you arrive at the same conclusion regarding its physical and structural integrity.
Okay.
So that is why you need to use several techniques which help us to confirm that the protein is structurally well formed.
Okay.
And finally, once this structural confirmation is done, you go for its functional analysis using various cellular based assays. And once cellular based assays confirm the integrity of the protein being functionally well, then you go for regulatory approval. So
Okay.
during this physical and chemical characterization, what we propose that it is obviously very important to do characterization of the purified protein as such, to know whether it is formed well. But once you formulate it using several other buffer components, and it is ready for being tested for various animal trials. You can use various innovative strategies which will help to characterize in a very quicker and an economical way.
Okay.
There is no need to do again these sets of techniques which I just mentioned
Mhm.
all over again after it is being formulated. So once it is formulated with various buffer components, you can simply do a quick check using a technique which will not compromise the characterization ability and also save the time and money which is being invested all over again for characterization. So, so instead of doing that, conventional characterization, we have uh, introduced the use of another technique like small angle X-ray scattering. So, in this technique, what happens is your protein molecule is bombarded with X-rays, and the X-rays, when they are bombarded on the protein molecule, they scatter the X-rays in various directions,
Uh-huh.
and these scattered X-rays give information about its size and its structure.
Right.
So, in one analysis, you can find out the information of its size, you can find out the information of its structure, and immediately pass that particular batch of biopharmaceutical drug for the next step instead of doing so many techniques altogether. So, this not only saves time,
Hmm.
this also makes the process economic.
Okay.
So, why economical? Because as you know that we are ourselves a developing nation,
Right.
and uh, although we say that biosimilars are very affordable treatment and as compared to their biological drugs which are the innovator drugs which were formed initially and now their replicas are called as biosimilars, although we expect them to be cheaper as compared to their innovator drugs, many people in our country are not able to afford them.
Yes.
So we need various strategies which further reduce the cost of these biopharmaceutical drugs, and therefore make this treatment accessible to people across socio-economic strata of the society.
Hmm. So, I wanted to ask, can biosimilars be correlated to how generic drugs are in the market? How generic drugs are copies of their synthetic drugs. And similarly, biosimilars are not exact copy but highly similar to their original biopharmaceutical drug, right?
Yes, we can make that analogy.
Yeah, okay. Mhm. And so monoclonal antibodies, they are one of the biopharmaceutical drugs, right?
Yes, they can be peptides, antibody drug conjugates, or bispecific antibodies, like a single antibody directed towards two different targets. So
Hmm.
there are several formats of maps which are being now made to improvise their function. The next part of the work is about improvising the function.
Okay.
So generally people try to perturb the structure of the antibody in some way or the other
Hmm.
in order to make it functionally better than its existing original structure.
Hmm.
So what they do is the monoclonal antibodies have basically sugar molecules in one of their region.
Hmm.
Now there is one particular sugar called as fucose. Now, if you remove fucose sugar from that particular sugar group, it is observed that the function of the antibody increases several fold, and it's a very well established fact.
Hmm.
But although this is very beneficial in case of some antibodies, in case of other antibodies where you see that the removal of fucose not only improves the function but also it causes several side effects because of the removal of fucose. So what do we do in the case of those antibodies where removal of fucose is causing additional problems? Therefore, what we propose that
Hmm.
instead of going for any structural modification, why not use the antibody in its native structure and use additional molecules along with it so that the function is improved, and also the structural disturbances don't cause any additional side
Side
effects.
effect.
Because the whole purpose of the MAP therapy is what? To have the medicine directly hit the target cell, that is a cancerous cell, or
Hmm.
any other target cell, which is a disease cell. In the case of traditional cancer therapy like chemo and radiation, this site directed uh, you know treatment is not achievable, and we see that cancer patients tend to lose their appetite, or they lose their hair, and
Huh.
several other side effects are observed. So although I don't make a statement that the protein therapy is completely devoid of side effects, but the side effects are very much minimum as compared to chemo and radiation.
Hmm, hmm, hmm. Got it.
So that is about the PhD work which is being carried out at NRG.
Okay. So basically, you said that for characterization, they are using a bunch of techniques that you mentioned.
Hmm.
But as an alternative, you're providing or you're working on an independent and separate, technique that would give the same information that those techniques give, right?
Yes, yes.
Okay. So is this the reason that monoclonal antibody therapies are costly? Because in India, the rituximab monoclonal antibody, it costs around one injection costs around 37,000 rupees,
Yes,
which is
yes.
very very high. And
So that is one of the steps of production of a biopharmaceutical product which can be focused to reduce the price. You can find out various strategies during downstream processing where
hmm,
you can find out innovative ways to purify the protein, or you can find out innovative strategies during the upstream processing. So not just characterization, but every step during the development needs to come up with new strategies which are cost effective and therefore mitigate the price of the drug.
okay, mhm.
So this work basically focus on characterization.
Right, got it.
Also, since my background is from biophysics, I was more comfortable with this particular step during the whole process of biopharmaceutical manufacturing.
Acha.
So that is another reason to choose this step.
Got it. Nice. This is quite exciting and interesting for me. Yeah. Now moving from the research topics and the research questions, I wanted to ask you, what do you like to do apart from research?
Apart from research, so I'm mostly into any fitness related hobbies.
Oh.
So I run marathons or cycling. I regularly work out too because I feel many PhD students tend to neglect their physical health over the period of time during as they reach the seniority.
Mm-hmm.
So I feel that it is very important to focus on your physical health along with doing your work regularly, and it really keeps you charged for doing your PhD work too.
Yeah.
So yes, that is one thing which I do regularly.
Okay. So, what is one advice that you'd want to give to the students who are aiming to do research in India?
Okay, so as far as research work is concerned, one thing which everyone should keep in mind that it is not that if you have cleared an entrance you would really do well in research. You shouldn't really correlate the ability to clear entrance with ability to do research, because you are clearing an exam on the basis of your theoretical knowledge. Here you are actually working. So you need to really like do some self analysis while you carry out some research work during your bachelor's or master's, and do a self critical assessment that whether you would be able to do something more complex than what you're already doing during your masters or your bachelor's. So once you have done that self assessment, you should really go forward for appearing interviews and so on. And although people prefer going abroad for PhD interview, for PhD and so on, even if you are not being able to go abroad for various reasons. We have fair amount of good research labs in India which carry out really good research. And I mean, if you are not able to go abroad, then you even have an equally strenuous procedure here where you have to appear for a panel of 30-40 people for 30 minutes, and appear for those interviews. So it is equally challenging to get a seat here as well. So don't really consider that if somebody is going abroad it is going to be better than what you are doing. It's all about how you do your work, because at the end PhD is mostly dependent on how you do it, and your guide is just going to be a support to not do something, you just not lose your track while you achieve your objectives. So it is largely going to be your contribution. So whether you go in the US or you stay in India, you are going to be you. It is just that the support is going to vary. So that is one thing I would like to advise. And the next thing is that not to be really disheartened by the negative results. I mean, we were taught during our bachelor's that there is nothing like negative results. If even if I refer back my bachelor's journal, like regular microbiology journal, my observation would run in pages, because we were asked to report what we got during the practicals. It was not like if my neighboring table has got a good results and I have not got that good result. Generally what teachers suggest is, okay, you report that another person's experiment result because they have got a proper result, because this can't be reported in journal. This practice was not followed thankfully during my bachelor's, and they really asked us to interpret what went wrong during my own experiment. So that is how my observation and conclusion would run in pages, and why it went wrong. So if you have that ability, like to tell people as to what is the other way of looking at this negative result, which and I mean quote unquote negative result, which is a very famous term. If you are able to work around it, you're just close to your quote unquote positive result.
Yes, exactly.
So yes, yeah, that are the two advices I would like to give.
That's awesome. That's awesome. Nice. Yeah, I'm in love with your research because not only it's interesting and exciting, but also the fact that any significant output from your research would directly benefit the people suffering from conditions like cancer.
Yes, yes. I mean, it is very much the need to have affordability factor in, while you pursue research, because many a times we carry out really good research, but it doesn't reach the required masses.
Exactly, exactly. So I love that fact about your research.
Thank you so much.
I wish you the best of luck for your PhD. And it was so good talking to you. And thank you for taking out time on a Saturday.
Oh, I mean, Saturday, I'm not still very used to with the weekend culture yet. So Saturday is very much a working day.
Anyway, but yeah, thank you for taking your time.
Thank you very much, Parth. It was great talking to you.
Thank you everyone for listening. Stay tuned for more exciting conversations.